Thank you.
So just a few minutes while people jump on. We love seeing how many people have joined us this afternoon. Going to give it just another minute.
Okay, we'll go ahead and get started. I'd like to welcome everyone back. Hope you had a nice little quick 15-20 minute break. We're going to have the pleasure of hearing from amolyt and their team. The two speakers that they have presenting today are Michael Kohler and Michelle Ovies. And with that, I will go ahead and turn it over to Michael, I believe.
Okay, thank you very much. It's a pleasure to be here with you today. I'm Mike Kohler. I'm the Chief Scientific Officer for amolyt Pharma. I'd like to start today by giving you a brief introduction to amolyt Pharma and then an introduction to AZP3601, our PTH analog, which is designed specifically for the needs of patients with hypoparathyroidism. And there we go. So first, amolyt Pharma is a private biotech that is dedicated to finding novel therapeutic solutions to rare endocrine diseases. Our mission statement is that we are building on our team's established expertise in therapeutic peptides to transform the lives of patients suffering from rare endocrine and related diseases. To accomplish this, we're relying on our highly dedicated and expert staff, led by a well-seasoned management team with broad experience in drug discovery and drug development, especially with regard to peptide therapeutics and endocrine disease, and who have successfully brought several drugs to approval and to the market that are now actively helping patients. We're a transatlantic company with offices and operations in both Lyon, France, and Boston, Massachusetts in the US, giving us the ability to take advantage of opportunities on both continents. Our current pipeline has 3 programs in it: AZP3601, which is for the treatment of hypoparathyroidism and about which you'll hear quite a bit more today. We also have AZP3813, which is a small peptide antagonist of the growth hormone receptor and will be used for the treatment of acromegaly. And we have AZP3404, which is an interesting small peptide which is based on the biology of leptin and insulin-like growth factor binding protein 2. And we are currently undertaking a number of pharmacology studies in order to determine the best lead indication for this new compound. So I'd like to introduce you now to AZP3601, our PTH analog for treatment of hypoparathyroidism. AZP3601 was specifically designed for the treatment of hypoparathyroidism in the laboratories of Tom Gardella and Michael Manstead at Massachusetts General Hospital in Boston. When they started this program, they set out with the goal of devising a PTH analog that could control the symptoms and improve the quality of life for hypoparathyroid patients by inducing a stable normalization of serum blood calcium levels that could be maintained over a full 24-hour period. In order to achieve this, you need to achieve a sustained effect on the reabsorption of calcium by the kidney, and as an additional important safety feature, to maintain bone integrity by having a neutral effect on the bone. To reach this goal, the MGH team examined how various PTH analogs interact with the PTH receptor and found that different analogs produce different conformational changes in the receptor and bring about different biological responses. To show you how this works, natural PTH binds to the PTH receptor and causes a change in the receptor shape. This is known as the RG conformation. As a result of that, a signal is launched inside the cell, which ultimately results in the triggering of the biological response. For example, an increase in blood calcium levels. As soon as the signal starts, natural PTH is released from its receptor and the signaling stops, and so too does the biological response. So once you've eliminated the natural PTH from the bloodstream, the response is over. It's a very short-lived response. And you can see in this graph where natural PTH has been injected into mice which have had their parathyroid glands surgically removed to create a model of hypoparathyroidism. And you can see the effects on blood calcium over a period of time after the injection. Compared to placebo, you can see that natural PTH 1 to 34, it does induce a significant increase in blood calcium levels. But once the PTH is eliminated from the bloodstream, the effect is over, and you can see that the calcium levels rapidly fall again back to baseline. So you need continuous receptor exposure or activation in order to maintain the signal and the response. AZP3601 has quite a different interaction with the receptor. It too binds to the PTH receptor and causes a change in the receptor shape, but this is known as the R0 conformation. It too launches a signal inside the cell, which starts the biological response. But once the signal starts, AZP3601 remains bound to the receptor. And as a consequence, the signal continues on, as so too does the biological response. So with only a very brief receptor exposure, you can initiate and maintain the biological response for an extended period of time. And here you see the same model that we showed you before with a single injection of AZP3601, and you can see that it induces a significant increase in serum calcium levels, which are maintained well past 24 hours after the injection. So therefore, the response is continuing long after 3601 is gone from the bloodstream. You can see that more clearly in this study, in which we injected AZP3601 into non-human primates, cynomolgus monkeys. On the left-hand side, you can see the actual measurement of AZP3601 in the bloodstream, and you can see that within 1 hour, the drug is completely eliminated from the bloodstream. However, as you can see on the right, the biological response, the effect on elevating serum calcium, is maintained for over 48 hours, with the peak occurring at about 24 hours after injection. So a very, very long, prolonged effect with a very short duration of exposure of the receptor and a very short half-life in the blood. Another interesting feature, and we do not have direct evidence for, but there is indirect evidence that activation of the R0 receptor appears to be preferential in certain tissues, most notably the kidney. We've injected labeled AZP3601 into animals and observed that it minimally and only very transiently binds to most of the known PTH responsive tissues, including the bone. However, it remains bound to the kidney for very prolonged periods of time, well past 48 hours. So this preference for the kidney and this prolonged action may contribute to the goal of an ideal treatment for hypoparathyroidism in terms of both efficacy and safety. In terms of efficacy, there's multiple mechanisms through the kidney where we can affect and normalize serum calcium levels. For example, we can increase resorption of calcium from the urine and return it to the circulation. There's an increased elimination of phosphate, and this is important because phosphate and calcium can combine in the circulation to form a product which is insoluble. So when we eliminate the phosphate, it frees up the additional calcium to be available in the bloodstream. And of course, it's well known that PTH stimulates the increased production of active vitamin D through the kidney, which is necessary for the absorption of calcium from the diet. In terms of safety, as a result of the increased resorption of calcium from the urine and you stop the continual flushing of excess calcium through the kidneys, we expect to have a decreased risk of kidney disease in the long term. The increased elimination of phosphate and therefore the decreased calcium phosphate product will be important for prevention of mineral deposits that occur in soft tissues, such as the formation of kidney stones in the kidney. And we have a natural effect on bone, which is important for the prevention of bone loss, which is a significant factor for all patient populations, and especially for hypoparathyroid patients. So AZP3601 601 is potentially differentiated as a treatment for treating hypoparathyroidism. The goals are to control symptoms and improve the quality of life for patients by maintaining stable serum calcium levels within a normal range over a full 24-hour period. In order to accomplish this, or I should say, with this goal accomplished, the patients can eliminate the need for calcium and vitamin D supplementation. And in order to accomplish these goals, we need to reduce the urinary excretion of calcium. And of course, as an added safety feature, we want to maintain bone integrity by having a neutral effect on bone. And we've been able to demonstrate a number of these parameters already in multiple preclinical animal models, and we are currently confirming them in clinical studies in normal volunteers, as well as in hypoparathyroid patients. And my colleague, Michel Ovies, will now take you through some of the results from those studies. So Michel, and I will stop sharing and allow Michel to share his slides.
Good afternoon, ladies and gentlemen. I'm very happy to be here with you this afternoon. My name is Michel Ovies. I am a medical doctor and I'm senior medical director at Amolyt Pharma. And I will now share with you, as Mike just said, the first result of our phase 1 clinical trial with AZP3601. As you know, hypoparathyroidism is considered as a rare disease, but you can quickly see on this number on the slide that a lot of people around the world are suffering from hypopara, and they are suffering all their life through. And for decades, conventional therapy has aimed at short-term symptom management with large dose of oral calcium and oral vitamin D. But I would like to make a couple of points regarding this. Despite this conventional treatment, you all know that blood calcium is not within the normal range over 24 hours in most of the cases. And this is responsible for a lot of symptoms you all know better than I do. And this is true for many hours of the day, of the 24-hour day. The second aspect of this conventional treatment with active vitamin D and with calcium is that in some cases, it might increase the urinary calcium excretion. Then you know that in the long term, excess calcium in urine may damage the kidney. And on average, there is a 4 to 5-fold increase in the risk of developing kidney disease when you are treated by this type of treatment for hypoparathyroidism. And this is an important point. The second point I would like to make before going into the data of our clinical trial is that you are not a homogeneous population. As you know, hypopara patients are different, and this difference ought to be taken into account when designing a treatment for this disease. And a few examples: some of you suffer severe symptoms that sometimes bring them to the hospital, whether other patients do have less symptoms. Some of you, for some reason, have some fragility of the bone beside hypoparathyroidism, like osteopenia or osteoporosis, and we're going to talk about this later. And also some of you already have kidney disease for additional reasons, like, for example, being diabetic or having hypertension that may both damage the kidney beside being a hypopara patient. And of course, a lot of hypopara patients, you know, are women, and postmenopausal women may also have reasons, and we're going to talk about it, to have a bone fragility. And the general point here is to say that this population is not homogeneous, and we ought to take into account all these factors when designing a treatment for your disease. Now, what about our clinical trials? This phase 1 clinical trials is divided in 3 parts: Part A, Part B, and Part C, as you can see on the screen. Part A and Part B have been designed and performed and completed in healthy volunteers, not in hypopara patients, while Part C, which is ongoing, is performed in hypopara patients. Part A is what we call a single ascending dose study. It has been performed in 52 healthy volunteers, while Part B is what we call a multiple ascending dose. It has been performed in 50 healthy volunteers for 2 weeks. What is it? Part A, in part A, we inject only one single injection of AZP3601 subcutaneously in the abdomen and assess the safety of this injection, and of course the efficacy as measured by serum calcium, urinary calcium, and bone biomarkers. While in part B, we not only inject AZP3601, but for 14 consecutive days, and for these 2 weeks, we assess the same parameters as in part A. Part C is midway currently, and uses, if I can say so, is performed in hypopara patient. 24 hypopara patients will be included in that study, which is composed of 2 parts, a first 4-week part called the main part of treatment, plus an extension period of 2 months. It's also for assessing the safety and tolerability, of course, of ZP3601. And of course, here we are in real life of hypopara patient, and we look at, as you know, serum calcium, ability to eliminate calcium and vitamin D supplementation for your treatment. We look at urinary calcium, and we look at bone biomarkers. So as Mike said before, the goals of our research program is to be able to control the symptoms and improve your quality of life, to maintain stable serum calcium within the normal range over full 24 hours, and this is very important, to be able to stop the conventional treatment, that is eliminate calcium and vitamin D supplementation, to reduce urinary calcium excretion, and to maintain bone integrity. So now let's go to the results of the first part A, which is the single ascending dose study in healthy volunteers. In this case, the first, of course, objective was to look at safety. And as we said before, more than 50 healthy volunteers have been treated with different doses of our compound, and there has been absolutely no safety concern. Some healthy volunteers had adverse events like nausea, headache, and somnolence for the high dose we have used, but they're transient and they were related to the rise in calcium. This type of subject are not used to have like you do in your daily life. So we consider this study as perfectly safe. A few results regarding the single ascending dose. These figures present to you what we call pharmacokinetic. It's a strange word to say that we measure the blood content of our drug, AZP3601, for 4 hours as presented on the y-axis. So if you look at the scale on the left part of the figure, it goes from 0 to 70 picogram per mil of our compound AZP3601 in your blood. And you see that different group of patients, usually 8 patients, receive 10 microgram for the first group, 20 microgram, 60 microgram, 90 microgram, and 120 microgram for the last group. If you look at the curve and the different part, you see, if you look at the blue curve, which is the most visible, the higher dose of AZP3601, you can see 2 important things. The first is the very rapid rise in the blood content of AZP3601 from 0 to near 50 in a few minutes, in 4 to 5 minutes, and then a gradual decrease down to 0. And this is true for all the dose we have used, and there is a dose dependence, meaning that the higher the dose, the higher the peak. But in all cases, you can see, and this is very important, that after 2 hours, there's nearly nothing left in the blood of AZP3601, as Mike Color presented previously in the animal models. So we said that this drug has a short half-life. She stays very short time in your blood. She circulates a very short time in your blood. And this is very important to remember. The second aspect is obviously what you are interested in. This is the measurement of your serum calcium over time. If you focus on the left panel, you see that we present here in different groups, as presented before, the concentration, the amount of serum calcium in your blood over 24 hours. As you see, in each color represents 1 dose and 1 group of healthy volunteers. You have different points at 0, at 6 hours, 12 hours, 18, 24 hours. And once again, you can see that there is what we call the dose dependence, meaning that the higher the dose we inject, the higher the serum calcium. This is the first point. The second point is that this rise in serum calcium is comes pretty early after the single administration in the morning of SZP-3601. A couple of hours, and then at 6 hours is already quite visible in all groups. But then there is a difference between the group. If you look at the yellow or orange curve, this increase does not last up to 24 hours. While for the high dose in red, in green, and in blue, 60, 90, and 120 microgram, you have the persistent high serum calcium at 24 hours. And this is very important. And I can tell you, even though it is not shown on this slide, that it takes 48 to 72 hours for the higher dose to come back to near baseline. In other words, I would like to make the point here that although the drug stays only 2 hours on average in your blood, the effect, as was mentioned previously by Mike Köhler, is very, very much prolonged and goes, in this case, more than 24 hours for the 3 high dose. And this is very important to remember. Now, we looked also at endogenous PTH. Of course, you know that these subjects are healthy volunteers. They have a normal secretion of PTH, unlike you. And it is important in this case to look at the what we call the kinetic, what happens to the secretion, the production of endogenous PTH in the blood of these healthy volunteers. And in fact, you see exactly the reverse of what you have seen before for serum calcium. That is, let's say 6 hours after administration of the drug, you see that there is a major decrease in serum endogenous PTH in that subject, which is very important for the 3 high dose in red, in green, and in blue. And the other aspect is that if you look at the scale of the time on the x-axis, it goes to 72 hours after one single injection, meaning once again that our drug, after a single injection, has a very prolonged effect, in this case on endogenous PTH secretion. Now, these were the result for the first study, the main result for the first study. Now, we're going to look at what we call the multiple ascending dose study. In this case, the healthy volunteers received one single injection in the morning of SZP-3601, but this was repeated every day for 14 consecutive days. In terms of safety, we had, as we had before, no safety concern. The administration was generally well tolerated, no severe or no serious adverse events in this case, and the subjects were perfectly fine. The most important result is presented here. If you focused first on the left panel of the figure, you can see that we used 4 different doses in this part B of the study: 20 microgram, 40 microgram, 60 microgram, 80 microgram. There were also 8 to 10 subjects per dose. And we here have the evolution of serum calcium. What do we see? We see in gray the placebo group, and you see there is almost no change as expected because they did not receive any amount of SZP-3601. The lower dose, 20 microgram in yellow, was not having a major effect or no effect at all when you compare to the gray line. But starting at 40 and then 60 and then in green 80 microgram, you see a strong difference in serum calcium, which increases progressively up to a quite high value in the green group, the 80 microgram group. What does that mean? It means that we also have for 14 consecutive days the same effect we have for 1 day in the single ascending dose study. But on top of that, we have new information. You can see that it takes 2 to 3 days to get at the plateau, I would say, of each dose, which is an important information. And if you look at the point 14, that is the day we stop the treatment, it takes also 2 to 3 days to come back in each dose to the near baseline value. So this is an important information. Our drug is acting quite quickly. And the second aspect is that between day 3 and day 14, we have a pretty steady value of serum calcium. And this is even more evident if you look at the right panel. On the top figure, we looked at serum calcium on day 7. And on the bottom figure on the right, we look at the same thing on day 14. And you see that when you increase the dose, you have a higher serum calcium value. And on top of that, and this is very, very important, you see that all the curves are flat. That is, the serum calcium is maintained for the 24-hour duration after the morning administration, suggesting that we have a very steady and stable calcium concentration in your blood. Now, we looked also at endogenous PTH, and you can also see that what we have seen in the single ascending dose study is equally true in the 14 consecutive day study. Endogenous PTH in these healthy volunteers goes down on day 1 and stays down until the treatment stops on day 14, and then comes back to near normal in a couple or 3 days, or even more for the highest dose. So we have a very consistent mechanism of action and results between single ascending dose and multiple ascending dose. In this part of the study, we also looked at serum phosphorus. Of course, serum calcium is your main objective, but serum phosphorus is also something that is important for hypoparathyroidism. And if you look at the left panel, you see that, as we already said, there is a dose-dependent increase in serum calcium when you increase the dose of AZP-601. But in the meantime, this study shows that we reduce serum phosphorus, which is a good thing, because we know that in hypoparathyroidism, there is a tendency to accumulate and to have too much serum phosphate, and which can be damageable in the long term. So this is a very good news for the future, although it's in healthy volunteers. Now, I would like to focus to finish my presentation on 2 important target organs, which are the bone and the kidney. Let's talk about the kidney first. Quite a lot of hypopara patients have chronic kidney disease. And as I said before, there is a 4 to 5 time-fold risk of having this disease in your case. And this is a very important point, which is in fact quite visible on this slide from a recent review by Gosmanov and colleagues, where the authors present the percent of hypopara patient with chronic kidney disease, and as compared to a non-hypopara patient, which are depicted as a gray line, versus the hypopara line, which is the blue line. What do you see is that over a 5-year period of time and follow-up, there is a much steeper increase in the percentage of patients that develop chronic kidney disease if you are hypopara than if you are non-hypopara patient, confirming that the kidney is a target organ that we must take into account this when we develop our drug. And this is what we did, in fact, in that phase 1 study. We looked also in healthy volunteers, the urinary excretion of calcium, and specifically one measurement that we call fractional excretion of calcium, which is basically measures the amount of calcium that is excreted in the urine or in the kidney. And what you see in the left panel is that whatever the dose that has been injected of AZP-601 to these patients, there has been certainly no increase in the excretion of calcium in these healthy volunteers, but even rather, in some cases, a decrease, which is a very good news. But more important, if you look at the right figure, we put on the same figure the evolution over the different doses on the x-axis of AZP-601, in orange of serum calcium and in blue of urinary calcium. And it is very evident that although we increase serum calcium in these healthy volunteers, we do not increase the urinary expression of calcium, which is very, very important because increasing urinary expression of calcium might, in the long term, be damageable to your kidney. Now let's talk a little bit about the bone issue. We said before that some of you, because of age, because they are menopause in cases of women, because they have, for other reasons, develop osteoporosis, they have some fragility of the bone beside having hypoparathyroidism. And of course, we know, as my colleague said before, that PTH analog reactivate what we call bone turnover. So we must take this into consideration when developing a treatment. And this is what we did by measuring the bone biomarker in our healthy volunteers study. What is it about? The hypoparathyroidism is sort of slowing down what we call the bone turnover. What does that mean? Our bone throughout the life is permanently sort of resorbing, that is destroying the old bone and building new bone, what we call resorption and formation. And these two parameters, CTX and P1NP, that we can measure in the blood, give an idea of the balance between the two phenomena, destruction and formation of bone. CTX giving an idea about the resorption, the destruction, and P1NP giving an idea about the formation of the bone. That must go together and being well balanced. And you see that in these healthy volunteers that have been treated for 14 days by AZP3601 at different doses, there has been no change, no detectable change in CTX or P1NP, suggesting that we haven't changed the balance and the equilibrium between these two phenomena, which is very important to keep the bone in, not to enhance the fragility of the bone in hypopara patients. So to sort of summarize this multiple ascending study, we've seen, first of all, that it's safe, and that we, as you have seen, we have a strong dependent effect on serum calcium, that this effect is sustained and stable all over the 24 hours, and that there is no increase, or in some cases a decrease, in urinary calcium excretion, and there is no whatsoever biomarker showing that there is non-balance in the bone turnover. So I would like to repeat the slide that was presented previously by Mike Köhler, saying that in our drug development, we aim at the 5 same things. We need, when we go to patients, to control the symptoms and improve the quality of life, to maintain stable serum calcium within the normal range over the full 24 hours, as we have seen in healthy volunteers. We hope to be able to eliminate calcium and vitamin D supplementation, to reduce urinary calcium excretion, and to maintain bone integrity. And with that, I would like to thank you for your attention, and Mike and I will be happy to take all your questions. Thank you very much.
Okay, it's unmuted. Go ahead.
All right, thank you so much. So let's go with a couple questions. Is it safe, beneficial for a hypopara patient with kidney failure to take this medication?
This is a very good, a very important question. It depends on what stage of renal failure we are talking about. You know, there are 5 stages of renal insufficiency, and down to which are measured by what we call the eGFR, which is an idea of how the kidney is able to do its job and eliminate different substances. And down to 30 ml per minute per 1.73 square meter of eGFR, there is no harm to treat with PTH analog hypoparathyroidism. It's even necessary. But after that, in my knowledge, there has been no studies showing that patient with very severe renal insufficiency, there might be, it might be feasible or not feasible, but safe to treat with a PTH analog. So most of the patients can be treated by a PTH analog, and these are the patients that are recruited in the trials in general.
Okay. All right, and that question was from Ariana. Thank you for that question. And how about Dasha asked, you measured endogenous PTH increase, but what if the patient can't produce their own?
Well, that's a good question. A patient, an hypopara patient, of course, can have can still have some PTH secretion, but I mean, this PTH secretion obviously is not sufficient because he wouldn't have hypoparathyroidism otherwise. And giving AZP3601 won't have any effect on this. AZP3601 is here to replace the missing PTH and do the job, and it won't have any effect on the remaining PTH secretion of this patient.
Okay, very good. And then Mindy wrote, why would you have high phosphorus, low urine calcium, and low serum calcium? What is considered having high phosphorus levels long-term, above 6 or 7, and is that concerning?
For phosphorus, I mean, having for the long-term high level in the blood can be harmful. Why? Because there are many reasons. As you know, calcium and phosphorus get together to build stones. And these stones can happen, especially in the kidney, can happen even in the brain, and can also happen in your blood vessels. And this comes slowly, but if it lasts for many, many years, it can damage kidney, for example, or lead to cardiovascular diseases. And so if you have a long-term hyperphosphoремia, this can be damaging all these organs and your good health. The value of serum phosphorus depends on the lab value you are using, but there is no, I would say, strict dependence, but the higher the phosphorus in the blood, the higher the risk of developing these diseases.
Okay, thank you. And then Ariana also wrote, how many times daily were the calcium levels tested during the study? And what is the GFR below which this medication has not been studied?
So as you have seen both in the SAD and in the MAD studies in healthy volunteers, we measured serum calcium 30 minutes after injection, 1 hour, 2 hours, 4 hours, 6 hours, 12 hours, 18 hours, 24 hours, and in some cases 48 and 72 hours, so very frequently. This was done not every day for 14 days. It was done on day 1, on day 7, and day 14. For the other days, we only measured in the morning, immediately before the administration of AZP3601. So for these 3 days, we have a lot of measurement which help to build the curve you have seen in my slides.
Okay. And then from Rita, she says, how do you account for the short half-life with prolonged activity?
Maybe
I can address this. As we were talking before, actually, AZP3601 has almost the exact same circulating half-life as natural PTH. Both of them are eliminated very, very quickly from the bloodstream. The difference is in how they interact with the PTH receptor. Natural PTH, as we mentioned, induces the RG conformation. So and the interaction is only maintained, the activation of the receptor, as long as the PTH is in the bloodstream. So once the PTH is gone, the effect is gone. But with AZP3601, the activation of the receptor carries the signal for an extended period of time, as you've seen, well past 24 or 48 hours, depending on the dose and on the particular circumstances. So it's really the receptor interaction that's driving this very long effect with the short half-life.
Gotcha. Thank you so much. And then let's see, how about, is there a way to assess efficacy of your PTHRP that's better than blood and urine calcium levels?
Can you repeat, please, because I didn't get the question.
So is there a way to assess the efficacy of your PTHRP that's better than blood and urine calcium levels?
I think that these are the very usual parameters. Of course, the best way would be to rely on symptoms of the patient, and this is, of course, the first goal we have. That is, if a patient feels better after administration of AZP-0601, it will probably go with a better serum calcium levels, and this is for sure. As for urinary excretion of calcium, this is not related directly to symptoms, but it's mainly related to the risk of developing kidney disease in the long term. So there is no better way that I know to follow the treatment of a patient that measuring blood concentration of calcium and sometimes urinary calcium excretion.
Okay. And then Serpent said, hello, when does AZP-3601 will be on the market? And are you doing studies PH2 or 3 outside the USA?
So we are, as I've presented, we are in the phase 1 study, and we are treating hypopara patient, and we expect to complete this study during 2022. And then we'll have to design, not to design, but to perform an additional study that will be done in hypopara patient with different doses of our drug, probably, and compared to placebo, as is usually done in this case. And of course, the study will be performed in the US, it will be performed in Canada, will be performed in probably in Europe and in different countries other than that, probably. So it will be a worldwide trial.
Okay, very good, thank you. And then just one more, somebody was talking about they have autoimmune damage to their kidneys with a GFR of 17. They were just wondering, would they be a possible candidate for the medication? Or are there other factors that would go into that?
That's obviously an important question, because we were only talking about specific adult population, but we know that there are a lot of children suffering from hypoparathyroidism. But this type of study comes after we show in adults that we are efficient, and what this is called a pediatric plan of investigation. This will come in some time.
Great, thank you. All right, well, I think that does it for, one more came in. Kathy said, if the drug has a short half-life, like normal PTH, but has a longer effect, how is it that you require a daily injection of AZP-3601?
Because, as you've seen, it's the effect that we have on calcium that is maintained over 24 hours. So by administering the compound on a daily basis, that effect accumulates and forms a steady state. And that's what gives you the very stable level of calcium that's maintained through the 24 hours and over additional days. So you don't have the fluctuations that are occurring. It's a very nice, steady maintenance of the calcium level.
Great, perfect. Thank you so much. Very good. So I think that's it for the questions.
Yeah.
Okay.
Awesome. And again, we thank you for your time and all the work that you guys have put into this, and excited to see the next step in this evolution. And with that, our next next session will start in about 15 minutes, so at 5:15, and we'll see you then. And again, thank you, Blandine and Michael and Michelle, for the work that you and your team have done. Excellent job.
Thank you.
Thank you very much.
All right, we'll see you back here in 15 minutes.
Thanks.
Thank you.